Hello, my name is Marc de Meyer and I'm an entomologist specialized in the identification, taxonomy and systematics of African flies, in particular tephritid fruit flies. I work at the Royal Museum for Central Africa, which is based in Teveur, Belgium. This presentation deals with an introduction to the main fruit fly pest species found in Africa. It is part of a series developed within the framework of the FFF Fruit Fly Free project, supported by the Standards and Trade Development Facility. In this presentation, I will provide some general background on each of the main pest species, how to differentiate them from each other using morphological characters, and some general aspects regarding their distribution and host plant range. For characters on how to recognize fruit flies as a group or the main genera, I refer to the respective separate presentations. As indicated in the presentation on the main pest genera, there are 6 genera worldwide that comprise the majority of pest species. These are Anastropha, Rhagoletis, Cerathitis, Dacus, Bactrocera, and Zogodacus. Of these 6 genera, 4 of them have representatives in Africa, either indigenous ones or introduced ones. For each of them, we will present some of the species most frequently encountered and responsible for the main damage observed on fruits, including crops that are biologically fruits but considered commercially as vegetables, such as tomatoes, pumpkins, or eggplants. The ones that will be presented are listed herewith. For Cerathitis, it concerns the Mediterranean fruit fly Capitata, the Natal fruit fly Rosa, the Cape fruit fly Kilisi, and the Marula fruit fly Cossara. For Dacus, it concerns Dacus ciliatus, the Ethiopian fruit fly, the pumpkin fly Dacus bivitatus, and Dacus punctatifrons. For Bactrocera, it concerns the 2 invasive species, the Oriental fruit fly Bactrocera dorsalis and the peach fruit fly Bactrocera zonata. Finally, there's the melon fruit fly Zogodacus cucurbitae. Here we see a typical female of the Mediterranean fruit fly ovipositing on an orange. It has the main characteristics of a Cerathitis species, with a rather short body, the wing marked with spots and streaks at the base and yellow-brown bands. The main characteristics to differentiate it from the other main Cerathitis pest species relate to the combination of the following. The abdomen has a silvery colored transverse band along the margin of the 4th tergite, seen on the left image, where the band is indicated by a red arrow, and the wing banding, the anterior apical band and the discal band are separated from each other by a transparent gap. The gap is encircled red on the right image. Furthermore, the thorax in dorsal view, as shown here on the image at the left, is mainly black colored with silvery areas. The postpronotal lobe, encircled at the top, is pale with a medial black spot. The scutellum, encircled at the bottom, has 3 black apical markings which are fused together into one large black area. The male specimens have some additional characters. On the fronts, they have an orbital bristle, which is pointing forward, is black in color, and flattened at the tip into a square or kite-shaped structure. This is shown on the image at the right, indicated with a red arrow at the top. At the bottom, the forefemur is shown. One can see along its posterior part a dense bush of yellow-orange bristles. Both these characters are missing in female specimens. Cerathitis capitata is widespread throughout the whole region south of the Sahara. This map shows point localities where specimens of this species has been trapped over the years. one can see that it is present throughout the whole of the Afrotropical region, as well as on islands both in the Atlantic and in the Indian Ocean. The species has also been introduced in other parts of the world, including Northern Africa. In general, it is considered to be a species that thrives better in drier and warmer conditions. It is very polyphagous, with several hundreds of recorded host plants. For Africa, there are records from more than 100 different hosts, belonging to about 30 plant families. These include all major horticultural crops grown on the continent. For monitoring, several attractants can be used. As for other fruit flies, protein bait will attract both sexes, but mainly females. Specific male attractants are trimethylure and egolure. The next species is Cerathitis coursera, known as the marula fruit fly or mango fruit fly. Here you see a habitus image. As in Cerathitis capitata, it shows the same kind of general body shape and wing markings. The abdomen is identical to the one observed in Cerathitis capitata, also with the presence of a transverse silvery band as shown here on the left. The wing is also identical, except that the discal band and the anterior apical band are joined together. The area is encircled on the image on the right. This is contrary to Cerathitis capitata, where these were separated. Also, the thorax markings are different. In Cerathitis coursera, the general body color is yellow-orange to orange-brown rather than predominantly black. The postpronotal lobe also is pale with a black medial spot as in capitata. However, the apical markings on the scutellum consist of three distinctly separated black markings. They are not fused together. The characteristic feature of the male, shown here on the right, are also unlike Cerathitis capitata. The orbital bristles are the conventional pointed black bristles directed upwards without an appendage. The posterior side of the forefemur has only a few dispersed hairs and not a dense bush. The mango fruit fly is also widespread throughout the sub-Saharan region. There are, however, some general differences with Cerathitis capitata. Cerathitis coursera is absent from the southern part of South Africa. Its distribution here follows the known distribution of one of its major hosts, the marula tree. It is also absent from most islands adjacent to the African mainland. It does not occur outside the Afrotropical region. There are therefore no records from North Africa for this species. It is a polyphagous one, but the host range seems to be more restricted than in Cerathitis capitata. It appears to be found often on Anacardiacea. One of the main indigenous hosts is marula, and before the arrival of invasive species like the oriental fruit fly, it was the main indigenous pest species for mango. As attractants, protein bait will attract both sexes, but predominantly females. Known male attractants are terpenyl acetate and egolure. The Natal fruit fly, Cerathitis rosa, and the Cape fruit fly, Cerathitis kilisi, are presented here together as they are very similar to each other and females cannot even be unambiguously differentiated. Actually, till recently, both were considered as one and the same species, known under the name Cerathitis rosa. On the abdomen, in addition to the transverse silvery band, there's a dark transverse band on tergite 3, indicated here on the left image by the red arrow. The wing pattern is similar to the one observed in the other two precedent species, except that the wing bands are usually more extensively brown colored. The discal band and the anterior apical band are separated by a transparent gap encircled in red on the right image. This is similar to the marking observed in Ceratitis capitata. Contrary to the two previous species, the dark markings on the scutum are not as clearly defined spots or stripes, but rather darker streaks. The postpronotal lobe is completely palish and without a medial dark spot. The apical markings on the scutellum are present as three clearly separated black spots, similar to what we saw in Ceratitis coursera. In males, the orbital bristles and the bristles on the posterior part of the femur are also similar to what we observed in coursera. However, the bristles on the forefemur can be more darker in color. The males of both species have one additional feature. The mid-tibia is along its margin for a large part covered by a row of dark and rather broad bristles as seen here. The row resembles somewhat the shafts observed in a feather of a bird, and presence of such rows is usually referred to as feathering. The area of the actual tibia where the feathering occurs is darker colored. Subtle differences in this coloration will allow you to separate male rosa from male Ceratitis kilesi. On the left image, one sees the mid-tibia in rosa. Here the black area is obliquely ending anteriorly, but the black markings extend straight up to the margin of the tibia. See especially the area pointed out by the red arrow. If you compare this with the image on the right, you see that the black margin is bordered on both sides by an area that is still yellow colored. The black marking does not reach all the way to the margin of the tibia. This is typical for Ceratitis kilesi. An additional, although not always clear character, is that the mid-tibia in Ceratitis rosa is usually somewhat broader and a more stout general appearance than the one in kilesi, which is more slender shaped. Both species are restricted to southern and eastern Africa, from South Africa northwards to Kenya. Their occurrence is given on this map with blue dots marking the presence of only kilesi and red dots only rosa. White dots are representing female or other specimens that could not be differentiated, and the places where both species occur together, what we call sympatric occurrence, are indicated in orange. It is obvious that Ceratitis kilesi distribution is more extended to the south. Also, Ceratitis kilesi appears to be found on some islands in the Indian Ocean, Reunion and Mauritius, where the species was introduced. What exactly determines the different distribution ranges for both species is not yet fully understood, but we have some indications that temperature can play a role. This is obvious from studies in areas where both species can occur together, but where we make observations along an altitudinal transect. We see that at lower altitudes, rosa can be more prevalent, while at higher altitudes, kilesi dominates. There may also be differences in host range, but these are not yet known. Literature records for Ceratitis rosa may refer to either that species or Ceratitis kilesi, as most of these records were made prior to the recognition of Ceratitis kilesi. From the known or confirmed host, it is clear that both are polyphagous, attacking a wide range of unrelated plant hosts. But the exact range for each species still needs to be established. Both species can be attracted to protein baits, trimethlur and egalure. Let us now look at some of the major pest species in the genus Dacus. Here we will present first the Ethiopian fruit fly Dacus ciliatus, of which you see a habited image. It has all the main characteristics of a daytime fly with an elongated body, predominantly brown in color but with yellow markings. The wing has a black coastal band which is apically extended into a spot. Here you see a close-up of the wing in the top left corner. The coastal band is rather narrow and the apical spot is pronounced but not very large, indicated by the red arrow. In addition, there's a brown marking along cell BCU, the so-called anal streak. The thorax in lateral view, as shown on the image bottom left, is predominantly brown with some yellow markings. The red arrow points to the yellow-colored cataturgite. The anaturgite situated dorsally of this is not yellow, but has the same color as the rest of the thorax. The image on the right shows the thorax in dorsal view. Specific with regard to the other species presented here is that there are no longitudinal yellow stripes in the posterior part of the scutum. Yellow coloration is restricted to the transverse suture and the postpronotal lobes. Dacus ciliatus is widespread throughout the whole of the African continent, including drier areas, for example in parts of the Namib and the Sahara Desert. The species is also found in Central Asia and appears to be introduced there, although the exact invasion history has not been studied. Dacus ciliatus belongs to the group of cucurbit feeders within the daysines. It is reported from about 40 different cucurbit crops. Still, there are also some records from non-cucurbit hosts, so the host specialization appears to be not complete. Dacus ciliatus also belongs to a group of species that are all cucurbit feeders that are widespread on the continent and that are not easily differentiated. This group includes Dacus frontalis and Dacus vertebratus. Differentiation between these species is not straightforward, so one has to be careful when reporting this or related species. Dacus can be attracted by protein baits, but there is no specific lure. The next Dacus species is a pumpkin fly Dacus bivitatus, of which you see a habitus image here. It also has all the main characteristic of a dayside fly as the one we mentioned when showing Dacus ciliatus. What is directly apparent in this species is the large, broad coastal band running along the anterior part of the wing, as shown here in the top left image, indicated by the red arrow. This is contrary to the other species presented, where the coastal band is only present as a narrow line along the margin. Also, an anal streak is present as in the previous species. When looking at the thorax in lateral view, as shown in the image bottom left, the yellow markings at the posterior part of the thorax consist of two yellow markings that are joined. This is both the cataturgite and anaturgite, indicated by the two red arrows. When we look at the thorax in dorsal view, shown on the right, we see that there are three longitudinal yellow stripes in the posterior part of the scutum, one medial and a pair of lateral ones. A particular character to differentiate Dacus bivitatus from the other species in the group is the presence of small hairs called microtrichia that fill the main area of wing cell C. This cell is situated at the basal part of the wing along the anterior margin, encircled in red on the image below right. The detailed image, top left, shows these microtrichia. In other species, these are restricted to a small part of the cell along the anterior and distal margin and otherwise is completely hyaline. As the previous species, Dacus bivitatus is widespread throughout the African continent, but in contrast with Ciliatus, it is largely absent in the drier parts such as the Namib Desert or the Sahelian region. It is restricted to sub-Saharan Africa, with occurrence on some islands in the western part of the Indian Ocean. It also belongs to the group of cucurbit feeders within the daisines. The host range is more restricted than that of Dacus ciliatus, with about 30 confirmed hosts. Also for this species, there are a few restricted records from non-cucurbit hosts, but these are rare and most of them belong to the family Solanaceae. It can be attracted by protein baits and the males by Q-lure. Another often encountered species among Dacus is Dacus punctatifrons, for which you see a habitus image here. The thorax shows several similar characters also encountered in the previous species, in particular the presence of 3 longitudinal lines in dorsal view and both the catateurogoid colored yellow, as seen in the lateral view. However, on the wing, the coastal band is represented by a small, darker colored area along the anterior margin and with a small extension at the end. An anal streak is also present. In general, the wing shows more similarity with the first species Dacus ciliatus. Again, it is widely distributed throughout the whole of the African continent, but appears to be less abundant and is probably missing from particular areas in Southern Africa, the Horn of Africa, and the drier areas in Western Africa. It is a cucurbit feeder reported from a limited number of commercially grown and of wild cucurbits. Also, as with the previous species, both sexes can be attracted by protein bait and the male specifically by Q-lure. Related to Dacus is the genus Bactrocera. Differences between both have been explained in the separate webinar on recognition of the main fruit fly pest species. The best known Bactrocera in Africa and in other parts of the world is Bactrocera dorsalis or the oriental fruit fly. This is an Asian species that was introduced in Africa around 2003. First, it was reported as a new species named Bactrocera invadens. Afterwards, however, it was demonstrated that both entities are one and the same, and Bactrocera invadens is now considered a synonym of Bactrocera dorsalis. The wing, shown here on the left, has an anal streak. The coastal band is narrow along the anterior margin, but complete, and runs till the apical part of the wing, where it is not really widened into an anal spot. The dorsal view has 2 longitudinal lines along the lateral margins. Otherwise, the scutum is brown with distinct black markings. These can differ in extent, with the dorsal part of the scutum almost completely black in some specimens. The abdomen is mainly brown, but has a distinct medial longitudinal black stripe. Along the third segment, there is a transverse darker band of variable degree. Together they form what is called a T-shaped mark. It was reported from Africa for the first time in 2003 from Kenya. Since then, it is recorded from most regions on the continent except for Southern Africa and above the Sahara. It is also found on all the islands in the Western Ocean, except for the Seychelles. It is a very polyphagous species, reported from more than 70 host plants divided over 30 plant families. Several other records of host plants are known from its indigenous region in Asia. As attractants, protein baits can be used for both sexes, while methyl eugenol is a strong attractant for the males. Another Asian species that was introduced into Africa is the peach fruit fly or Bactrocera zonata, of which you see a habitus image here. It is very similar to the previous species, but can be differentiated by a few characters. Regarding the wing markings, the coastal band along the anterior margin of the wing is not continuous, but interrupted in the middle, indicated by the red arrow top right. The anal streak is also missing. The scutum in dorsal view shows two yellow longitudinal lines, but otherwise is uniformly brown without the distinct black markings as usually observed in Bactrocera dorsalis. Also, the T-shaped marking on the abdomen is less distinct. At most, there's a narrow longitudinal dark line in the middle, but without a clear connection to the broad transverse band. Currently, the distribution of Bactrocera zonata is limited to the northeastern part of the continent, with records from Egypt, Libya, and Sudan, as well as the Arabian Peninsula. It is also recorded from some of the islands in the Indian Ocean like Mauritius and Reunion. However, since Bactrocera dorsalis was introduced there, it seems to be outcompeted by the latter and is becoming rather rare on these islands. The host range in Africa for this species is still rather limited, with known records from about 10 different hosts. However, in its native range, it is known from a much larger range and the risk exists that it could expand it also here in Africa. The same attractants as for the oriental fruit fly can be used, protein bait and for the males methyl eugenol. Finally, there is one Zogodacus species we would like to present here. It is the melon fruit fly Zogodacus cucurbitae. It is also of Asian origin and was introduced in Africa. Previously it was known as Bactrocera cucurbitae, but as Zogodacus is now considered by most researchers as a separate genus and not a subgenus of Bactrocera, the proper scientific name is Zogodacus cucurbitae. Zogodacus cucurbitae can be easily recognized by the enlarged apical spot at the end of the coastal band in the wing, but also by the presence of an additional marking along the crossvein DMCU. Both of these characteristics are pointed out by the red arrows on the top left image. The scutum is mainly brownish colored and characterized by 3 longitudinal lines in the posterior part, 1 median and a pair of lateral ones. The abdomen shows a T-shaped marking, which is usually less pronounced than what we observed in the oriental fruit fly. Zogodacus cucurbitae was reported from the African mainland in the first half of the 20th century from Kenya and Tanzania, and for a long time the only known records were restricted to this part of the continent. Over the last 20 years, however, the species has been reported from a much larger geographical range, both in Western and Eastern Africa. It is also known from a number of the islands in the Western Indian Ocean. As the name implies, Zogodacus cucurbitae is mainly a cucurbit feeder, recorded from a wide range of different Cucurbitaceae, both wild and cultivated. However, it is regularly also recorded from other non-cucurbit hosts, although they are much rarer. Both species can be attracted by protein baits, while the males can be attracted by Q-lure, similar to what we observed for a number of Daco species. This concludes the session on the main pest species of fruit flies found in Africa. Evidently, this is only a small representation of the approximately 400 different fruit flies that are known to be frugivorous and the more than 30 species that are regularly encountered on different kinds of crops. However, presenting all of them would take us too far. It is recommended to consult comprehensive identification tools or a specialist taxonomist for identification confirmation. Many thanks for your attention. This presentation was created in collaboration with Stellenbosch University. We would like to thank the funding agencies as well as the individuals and institutions who in one way or another contributed to the actual development of the presentation.